So I counted. What I did was to detach them of this plate. I counted them to make sure that I had enough cells to do the experiment, and I plated them in. I wanted to plate them in 2 6-well plate, but I plated them finally in 1 6-well plate and in 1 12-well plate. I plated them in different wells, so I will be able to put them in contact with different conditions. After that, I would like to wait for them to adhere to the support. It will take maybe 3 hours, so we will leave the lab for today. And tomorrow I will come back and I will put different chemical inhibitors in contact with them to be then able to extract on Monday the RNA of the cells. And I hope that the same day I will be able to do a study of the genes that are linked with the inflammation and with the immunosuppressive phenotype that I showed you before, so M1 and M2. You remember what messenger RNA is, right?
Yeah.
So you have the cDNA, you have the DNA that is in the nucleus, and inside the nucleus, the DNA, inside the nucleus, you have proteins that bind the DNA, and that either forbids or triggers the reading of certain sequences of the DNA. And once the DNA is read, it is transcribed in mRNA, and then this mRNA will be able to produce proteins by transcription, but with the ribosomes.
Yeah.
And then these proteins will be folded into the ER, as I told you, and etc. So this is done. I want to keep that, so I'm going to label it. In fact, it contains a lot of a molecule that is called GM-CSF. GSF for colony-stimulating factor, and GM for granulocytes macrophages. And it's a molecule. It's a protein that induces the differentiation of macrophages. And so I think I can reuse it for other cells that I will have extracted from the bone marrow of the mouse.
Yeah.
To differentiate them into macrophages. I put it back.
And that is the incubator.
Yes.
Now you done for today?
Yeah, I'm done. To wash everything, because otherwise, as you know, there will be bacterial infections. When I have a bacterial infection, it takes time. In fact, it takes time to kill the cells. That's why it's difficult to know it, to detect it at first. The cells will start to grow more slowly. Their morphology will be different, and every experiment that you will start with them will be wrong. Okay, so this off, and I put it on UV to kill the bacteria. So put that back. You want to take a photo of the fridge? So you have to transfer the proteins on the membrane. Well, it's this experiment I will have to redo it tomorrow.
Okay.
This is a gel, but I'm not going to explain you. I think it's no use. So you have a protein ladder like that, exactly like this one. You do an electrophoretic migration before in a gel. So sorry, yeah, okay, I can explain you finally. You have a gel, and you have small wells in your gel.
Yeah.
And in each well you will load your sample.
Yeah.
And for 1 hour you will put current. You will put your gel in between 2 slices of glass, and you will put into a solution, and then you will do an electrophoretic migration, so that your proteins will migrate from up to down the gel according to the molecular way. Then you need to transfer what is on your gel to a membrane, exactly like this one. And then you need to put in contact the membrane with several antibodies. So as you may know, an antibody detects specifically an antigen, and if you put a specific antibody, then you will be able to determine the expression of one of the antigen of your protein of interest. So for example, the protein of interest was here with JPDH, and you can see that this bond and this bond are more intense than this one, for example. And by that you can do a relative comparison of the expression of proteins from your samples. That's how it's done. We don't study directly the expression of the transmembrane proteins, because their expression is constant, in fact. The expression of these proteins are constant, but what is different is their activity. So by being active, they will activate other proteins, and these other proteins will bind on the DNA, and so they will induce a different expression of certain kind of mRNA. And by doing that, they will increase the production of certain proteins. So indirectly, you can see the activity of IL-1 alpha, PERK, and ATF6 by analyzing other proteins that are of the same pathway, we say. This time, instead of transferring it to a membrane, I let it overnight, and I waited, because it was to quantify the activity of an enzyme. This enzyme degraded the gel, because the gel was made of gelatin. And because of that, you can see that there are bands where there are nothing, just a white band. It's because the gel was degraded. So initially the gel is completely white, but I put a blue solution on it to stain it. And of course, because there are holes, there are lacunes, the gel cannot be stained, because there is a hole, of course. And that's why it's white.
And this is called what?
A gelatin zymography. I don't even use antibodies for this one. I have the protein ladder that is here to help me to determine what is at what molecular way I should watch my protein of interest. The two proteins that degraded the most gelatin were MMP9 and MMP2, according to the literature. I was not able to see any activity for MMP2, just for MMP9, which is of 92 kilodaltons, I think. And this is correct here, because this bond is 100 kilodalton and this one is 75 kilodalton. So the bond is between 100 and 75 in square runs, since it should be of 92 kilodaltons.